Now I want to demonstrate how we extract DNA of myxomycetes for barcoding in a 96 well scale. Therefore we have here these little plastic racks with 96 little holes, 96 wells, and these holes are made that these little 200 microliter tubes for DNA extractions will fit in. So all what we need is such a tube, a needle. I take a relatively blunt needle to carry over a bunch of spores with simply poking once into a sporocarp. What we need will be about 400 spores, not more. So let's take a specimen. You all know these kind of myxomycete specimens, and here you see the sporocarps in. I just put it under the dissecting microscope, but basically the whole procedure would as well work without a dissecting microscope. And now I will just try to get a sporocarp, or better a few spores from a sporocarp. So simply poking with a needle into the sporocarp would be okay. And then you see a couple of hundred spores which will cling to the tip of the needle. It's very hard to see. And then I put those into the tube, and on the ground of the tube I will move the needle a little bit around by just turning it gently, and that's all we need. So even if it is hard to see, we will later look under the dissecting microscope. You will have a couple of hundred spores in it, and that's all we need. Right now, please wipe the needle thoroughly with one of those paper towels. And that's very important, to have a clean surface of the needle again. A needle works better than a forceps since it's very easy to clean. So dispose the paper towel and you have a clean needle again. What is really important, just carry over only a couple of hundred spores. Don't put a whole sporocarp into the vial. It will be too much. There are inhibitory substances which will hamper the PCR reaction later. A few hundred spores, that's all we need for that kind of method. That are the spores. Here you see the couple of hundred spores that might be already been 1,000 in the bottom of the tube. That is all we need. Don't put a whole sporocarp in it.